Welcome to Hephzibah Simple Class. Today, I shall be talking on biosynthesis of glycogen. The biosynthesis of glycogen, also known as glycogenesis, is designated as the process of adding D-glucose molecules, one after the other, to a growing chain of glycogen. Glycogenesis or biosynthesis of glycogen, in another word, is the synthesis or formation of glycogen by the successive addition of D-glucose units to a primer and alpha 1 to 4 polyglucose chain containing about 4 to 7 glucose residues. The primer formation is initiated by glycogenin, a primer protein. The biosynthesis of glycogen may begin with a D-glucose or glucose 6-phosphate molecule, facilitated by enzymes and stimulated by insulin in response to high blood glucose levels. The enzymes involved in the synthesis of glycogen include 1. Glucokinase or hexokinase. 2. Phosphoglucosamidase. 3. Glucose 1-phosphate uridine or UDP glucose pyrophosphorylase. 4. Glycogen synthase. 5. Branching enzymes, also known as amylo 1,4 to 1,6 transglycosylase. However, glycogen is a multi-branched polysaccharide of D-glucose units that serves as an energy storage form in animals, fungi, and bacterial cells. Similarly, glycogen is a polysaccharide of D-glucose residues in which chains of the glucose residues are joined together by alpha 1 to 4 glycosidic linkages, and at every branch point found alpha 1 to 6 glycosidic linkages. Moreover, glycogen is a very large molecule containing over 1,000,000 D-glucose residues, with branching occurring at about 8 to 12 D-glucose residues. There is one reducing end per a macromolecule of glycogen, in which the terminal glucose unit is either unsubstituted at carbon 1 or linked to protein. All the other terminal glucose units are non-reducing with a hydroxy group at carbon 4, where a newly D-glucose unit is glycosidically linked with carbon 1. In humans, glycogen is formed and stored mainly in hepatic or liver cells and the skeletal muscles. In the liver, glycogen is essentially abundant, where it may attain up to 10% of the organ and serves as a short-term energy source by supplying glucose in response to low blood glucose levels. The skeletal muscle glycogen is about 1 to 2% of the muscle, providing a readily available glucose during exercise to support anaerobic and aerobic energy conversion pathways. Now, let us see the biosynthesis of glycogen pathway stepwisely. Note that the biosynthesis of glycogen occurs in the cytosol or cytoplasm. Step 1. Glucose is phosphorylated or converted to glucose 6-phosphate. The phosphorylation is catalyzed by glucokinase or hexokinase with the release of ADP from ATP. Step 2. Glucose 6-phosphate is epimerized or converted to glucose 1-phosphate. The epimerization is catalyzed by phosphoglucosamidase. Step 3. Glucose 1-phosphate is activated by uridine triphosphate UTP and converted to uridine diphosphate glucose UDP glucose and a pyrophosphate. This conversion is catalyzed by glucose 1-phosphate uridine or UDP glucose pyrophosphorylase. Step 4. To a primer polyglucose containing about 4 to 7 D-glucose residues initiated by glycogenin binds by glycogen synthesis, adding UDP glucose to the carbon-4 hydroxy group of the glycosyl residue on the non-reducing end of the growing glycogen chain. Step 5. Branches are made by glycogen branching enzymes, which transfers a new glucose residue onto the growing glycogen chain via alpha 1 to 6 glycosidic linkage. Here we come to the end of this class. I want to acknowledge and appreciate authors and owners of every image and material used in this